The cloning of a living organism is a concept that humans have had a fascination with for a very long time. Humans have wanted to see if it is even possible for one to create life identical to one that has been created before. These ideas are seen in books, folklore, and legends throughout history. It wasn't until 1996 that scientists were able to create a living mammal that was the exact replica of the organism that preceded it. What these scientists, Nguyen and Keith Campbell, are doing now is somatic cell nuclear transfer, which is replacing the nucleus of one cell with another. They are using the DNA from one sheep and copying it into the egg of another sheep, which will be the success that gets them a viable clone. But this process is not as easy as it currently looks. Why did you choose Campbell on this assignment?
Campbell's knowledge of cell cycle proved instrumental and directly correlated with advancing the technique of nuclear transfer developed by Rosalind.
You led the team that created Dolly the Sheep, right?
Yes, correct. My team, however, was originally focused on genetically modifying sheep for agricultural purposes to create genetically modified livestock. Our experiments were trying to cut down on the number of animals needed for research in agriculture. Eventually, my team and I became more interested in how we can perfect the sheep with the same qualities.
So, what is your experience with this topic?
In my early career, I studied cancer and the causes and mechanisms of it, as well as the cytoplasmic control of the nuclear behavior of amphibian cell structures. This gave me experience with cell growth, division, and embryos, which I was able to apply to the process of cloning.
What is your background with this topic?
Throughout my scientific career, I have been focused mainly on embryos and cell research. My contribution was mostly in the development and study of the eggs when they were in the embryonic stages.
What did you provide to the team in this endeavor of cloning the sheep?
I was able to apply my experience with nuclear cell transfer as well as my experience with microbiology and embryonic cells in creating a viable clone of the sheep.
So why did you end up cloning?
We ended up with cloning to look at how cells change during the developmental stages of animals. Dolly got all the big attention and is what people think of when we say that we cloned a sheep, but Dolly wasn't the first clone we made. Morag and Megan were other sheep we cloned, but were created from embryonic cells. Dolly was the most important, however, because she was cloned from an adult cell, which proved that animals could be created from adult cells of an organism, and not just embryonic cells. Creating viable clones for the experiment was a long process. It took extracting cytoplasm from an adult mammary gland cells in a way that would allow us to possibly extract DNA with it. When we got working ones, it was an accomplishment. When we revealed Dolly to the world in a paper that we published in the journal Nature, as we expected, there was a lot of controversy surrounding it. The college received nonstop phone calls on the topic from all over the world, ending up in a total of 3,000 calls. Some brought praise while others denounced that they were scared of the Pandora's box that this advancements could lead to. I myself was quite hopeful. This advancement could also lead to advancements in medicine and regeneration on a level that one couldn't possibly believe 20 years prior.
Cloning animals, in and of itself, wasn't really the problem. It was what it could lead to. When we weren't talking about the animals being modified for human use, it made sense for people to be a bit worried. The general gist of the argument against cloning is that it could lead to designer babies if we continue down the road with no regulation. Many birth defects.
On July 5th, 1996, Dolly was brought to life through multiple series of trial and error. Following her death, her life was filled with flashing cameras and continued controversies about cloning. Despite this, she managed to live an average life. She gave birth to 6 lambs before she was put to death in 2003. After her death, primates, mammals, and even endangered species have been cloned using the same techniques learned through Dolly's experimentation. Some scientists have even employed the use of somatic cell nuclear transfer to prevent the spread of mitochondrial diseases from mother to child. Overall, the test of Dolly has a vast impact on the scientific community. Without this, many scientific discoveries would not have been made.